will be recorded. Will be recorded, just a part of the talk, so the final questions and discussion will not. And so it's my great pleasure to introduce Nuno Silva, very focused and enthusiastic master student, I've been told, since we don't know each other personally, but Carolina and Antonio were very enthusiastic on recommending you for this talk. So Nuno is finishing his master degree that he did in CEDOC. His main interest, scientific interest, is inflammation, and he wishes to pursue a PhD in this topic. He has also published most of the work that he will be presenting today. So I'm very happy and very excited to see the work that he has been developing at CEDOC in this the drug screening project. So thanks a lot for accepting this, Nuno, and the stage is yours.
Okay, thank you. So let me just share the screen. Okay, are you seeing?
Yes.
Let me just get the pointer. Okay, so can I start it? Yes. So first of all, I would like to thank the invitation and also the opportunity to present this work that we have developed in the tissue repair and inflammation lab at CEDOC. And this work is entitled A Dietary Cholesterol-Based Intestinal Inflammation Assay for improving drug discovery on inflammatory bowel disease. Briefly, during this presentation, I will guide you through the development of this discovery platform. I will basically explain its advantages, and then I will present the screening that we already performed by using this discovery platform. And in the end, I will talk a bit about our future planned experiments and also the outcomes of this work. So inflammatory bowel diseases comprising Crohn's disease and ulcerative colitis are chronic inflammatory disorders of the gastrointestinal tract that can affect the patient's quality of life. Nowadays, IBD lacks a cure, and it affects almost 6.8 million people around the world. And because of the chronicity of this disease, it implies large costs to the society healthcare system, and it is also associated with high prevalence and also high incidence worldwide. And the problem is that this disease is already prevalent in industrialized countries, but in newly industrialized countries where this disease is not so prevalent yet, its incidence is rapidly increasing, and so soon it will constitute a growing health problem globally. And as examples of rapidly growing incidence of inflammatory bowel disease, we have, for example, China, which is a very populated country. Okay. And despite this increase in incidence, the onset of IBD is still poorly understood. Nowadays, it is thought to arise from a conjugation of multiple factors like aberrant immune responses, intestinal microbiota, genetic susceptibility, and also environmental insults. And this complex etiology of IBD makes the understanding of the disease very challenging, but it is also necessary if we want to develop effective therapies. Importantly, and regarding these environmental insults, it has been proposed that the human diet can play an important role in IBD. And interestingly, the spread of the Western type of diets, which are diets that are very rich in fat and cholesterol, has been suggested to contribute for a general increase of IBD incidence. And furthermore, a causal relationship between the increased consumption of high cholesterol diets and the increased risk of also developing IBD has also been suggested. And as previously mentioned, there is no cure for IBD, and the treatments that exist are used mainly to minimize symptoms and disease progression, and they constitute mainly immune system suppressors, antibiotics, and anti-inflammatory drugs like mesalazine and glucocorticoids. And the problem with these drugs is that they affect the body systemically, and so they have significant side effects all over the body. So basically, there is the need to find novel therapies for inflammatory bowel disease, and that was exactly the goal of this project. So to discover new therapeutic strategies for inflammatory bowel disease. Okay, and for that we used our favorite model system, the amazing zebrafish larvae. Because zebrafish larvae has several advantages to everyone that wants to study immunity and inflammation. First of all, it is a vertebrate model system that shows conservation of the most innate immune inflammatory pathways with the mammalians. Furthermore, it is optically translucent, so basically with the fluorescence microscopy you can look at your cells of interest at a cellular resolution. And to help with this task, there are several transgenics that highlight immune cells and also different tissues. And as examples, we have here 3 transgenic lines. Okay, one of these transgenic lines highlights neutrophils in red. Another one is a transgenic line that highlights the intestinal enterocytes with green. And another one is a transgenic line, is a double transgenic line that highlights both the neutrophils in red and the intestinal enterocytes in green. Okay. Furthermore, there are already developed models of IBD or acute gut inflammation that I will explore further in this presentation. And finally, they are outstanding models for screenings because to apply the compound to the fish and to test that compound, you just need to soak the fish in the water with your compound of interest, and then you see the desired effect that you want to assess. So to study inflammatory bowel disease and inflammation, first one needs to find a way of inducing intestinal inflammation in the zebrafish larvae. And there are several ways of doing so. And one of the methods consists on a chemical induction using chemical agents like TNBS and DSS. And these chemical agents are also heptanizing agents. And what they do is that they will promote intestinal damage in the zebrafish larvae. And this method is mainly used, is widely used as a fast, reliable, and also easy to handle methods. And it is used especially for drug screenings. Okay, and to do this, larvae are basically exposed to TNBS or DSS dissolved in the fish water for 3 days, and at the end of the assay, larvae present a robust and significant increase in the number of neutrophils in the intestinal epithelium, as you can see here in this image when compared to controls. Furthermore, recently, an high throughput and high content drug screening using zebrafish was performed in the context of IBD, and they aimed at identifying modifiers of intestinal neutrophilic inflammation. But however, the chemical treatments have led to 2 major drawbacks. First, it seemed to induce significant side effects, as overall neutrophilia was observed in the zebrafish larvae. As you can see here, a large number of neutrophils all over the body of the larvae. And second, TNBS and DSS also interfered with some of the screening compounds by inducing toxicity to the larvae or by changing the structure of the compounds that were being tested. And because of these, those compounds failed to be tested for their immune-modulatory properties in the intestine. So besides this chemical induction, there also exists an environmental and so a more physiological way of inducing intestinal inflammation. And this is done basically by feeding larvae with a high cholesterol diet. Okay, and briefly, this was done by Progatsky and colleagues, And they reported that after a 6 hours feeding period with a 10% high cholesterol diet, zebrafish larvae have an increased number of neutrophils in the intestinal epithelium when we compared with the control diets without cholesterol. And so basically they demonstrated an inflammation peak occurring at 18 hours post-feeding, and also a resolution phase that occurs 24 hours post-feeding. Furthermore, and by testing several inhibitors of the cholesterol uptake and of the inflammasome and the NF-κB signaling pathways, they basically demonstrated that this inflammatory process relies on the cholesterol uptake, the cholesterol binding via the Niemann-Pick C1-like protein that is present in the intestinal epithelial cells. And also, it is also dependent on the activation of the pattern recognition receptors and on NF-κB signaling. And what happens here is that briefly, the signal, this is the signal 2, the pattern recognition pathway, will lead to the production of pro-IL-1 beta in the intestinal enterocytes, and then signal 1 is responsible for the production of active IL-1 beta from the pro-IL-1 beta via caspase 1 mediated inflammasome activation. And then this active IL-1 beta is finally needed for the recruitment of neutrophils into the zebrafish intestine and to start and trigger this inflammatory process. Okay, so the work performed by Progatsky, from our point of view, is very interesting, but it is not suitable for screening purposes. Okay, but it is interesting because it allows us to suppress the drawbacks that are associated with the chemical induction of intestinal inflammation. We just need to optimize it. Okay, because as you can see, it has a small p-value with a high standard deviation and a considerably high sample size of about 30 larvae per time point, which is not suitable for a screening. So basically, we thought about optimizing this protocol reported by Progatsky and colleagues to try and develop an imaging-based discovery platform to identify modulators of intestinal inflammation that takes advantage of the transparency that we have in transgenic zebrafish larvae that highlight neutrophils. So very briefly, the idea was to induce intestinal inflammation in the larvae with a high cholesterol diet, okay, with the recruitment of neutrophils. Then we fix the larvae at the inflammation peak and perform a semi-automated imaging process to quantify the number of neutrophils that exist inside the larvae intestines. And this will be done resorting to the Zeiss Axio observer that we have here at ZDoc, which is an inverted wide field microscope that basically allows a semi-automated image acquisition, which is fairly suitable for screening purposes. And I have to say that this microscope reached ZDoc through congenital fundings, and it has facilitated my work by a lot. So, and indeed, we optimized the feeding strategy of the protocol by increasing the feeding time with a high cholesterol diet from 6 hours, which is the one used by Progatsky, to a 24 hours feeding period in transgenic larvae highlighting neutrophils. And this was done to understand which feeding time can give us a more robust result. After this feeding time, we let larvae without food for circa 15 hours, which is important so that the peak of inflammation actually occurs. And in fact, after these 15 hours, what we can observe is that there is a nice recruitment of immune cells to the intestine when larvae are fed with cholesterol when compared to normal food without cholesterol. And we can analyze this intestinal inflammation by calculating a neutrophilic inflammation index. Okay, and this is basically the area that is occupied by neutrophils within the intestine of the larvae, meaning that we calculate the area of the intestine here in green that is occupied by neutrophils that you can see in red. And as you can see, and also as an example, here we have a cholesterol-fed larvae with an index of 3% and a normal food-fed larvae with an index of 0.5%. So this is a scheme. This scheme is the image analysis pipeline of the neutrophilic inflammation index, and it is for you to have a better understanding of this analysis method. So basically, after image acquisition, we perform image deconvolution, because images are acquired in a wide field microscope, and deconvolution will allow us to have the cellular resolution of the neutrophil signal. Okay. After we perform the maximum intensity projection of both the bright field and the M-cherry channels, and with the bright field channel, we manually draw the intestine of each larvae, creating a region of interest. And in the M-cherry channel, we basically select the neutrophils, creating what we call a neutrophils mask that is performed by an intensity threshold. Okay. And in the end, we basically ask image J to calculate the neutrophils area fraction inside the intestine. So this is a fairly quick and semi-automated method that can be used to analyze screening data fairly rapidly. Okay. So by using this index, we can observe a nice increase of the neutrophilic inflammation index, respecting two controls when larvae are fed with high cholesterol diet for 12 and 24 hours. And with these results, we repeated the experiment to choose the best feeding time between the 12 and 24 hours feeding period. And what we found was that the 24 hours feeding period was giving us more robust results and with higher reproducibility between each experiment. And so we elected these 24-hour feeding periods with a high cholesterol diet to induce inflammation in our discovery platform. Okay. After that, and having the protocol to induce intestinal inflammation in the platform, we needed a positive control, meaning a drug that is known to abrogate high cholesterol diet induced inflammation. And for that, we tested the caspase-1 inhibitor, which basically halts the activity of caspase-1 to activate IL-1 beta. And thus, this will impair the recruitment of neutrophils and myeloid cells to the intestinal epithelium, and also impairs the trigger of inflammation. And as you can observe, caspase-1 inhibitor significantly decreased the inflammation promoted by the cholesterol, and almost to control levels. And so with these results, we elected the caspase-1 inhibitor as the positive control for the screening. And finally, and just to validate that our platform was an IBD-like discovery platform, what we did was to test two different anti-inflammatory drugs that are already used in the clinic for the treatment of inflammatory bowel disease, which are mesalazine and prednisolone. And what we observed is that the neutrophilic inflammation index is strongly reduced in the presence of these drugs. And this suggests that this platform can be used for IBD-related research. So this optimization led us to the development of a discovery platform that was recently published at Frontiers in Cell and Developmental Biology. Basically, we put around 15 larvae per well in 12 well plates. Then we have the compounds, and after we challenge larvae with HCD for 24 hours. After we change the medium and take out the food, but always in the presence of the compounds. And in the end of the 15 hours, where the inflammation peak occurs, we fix the larvae and perform a semi-automated image acquisition and also a semi-automated image processing and analysis to understand if the compounds can have an anti or pro-inflammatory profile. And what were actually the compounds that we were investigating? So together with Claudia Sanz and Diogo Carregosa from the Molecular Nutrition and Health Lab of SEDOC, we were interested in compounds coming from polyphenols. Okay, and these polyphenols are coming from fruits and vegetables, and they are well known to have an anti-inflammatory and also antioxidant properties. Okay, and interestingly, what is interesting about these polyphenols is that when they are ingested, they are subjected to digestive, colonic, and hepatic processing, and actually the molecules that are really available to go into the tissues or to circulation are the so-called low molecular weight polyphenol metabolites. And so most possibly the reported benefits of polyphenols are actually due to these metabolites. So what we did was to test their anti-inflammatory properties in a screening platform. And so this is the screening that we performed with those metabolites, where we have already screened 29 of the metabolites. And in the screening, we used two different concentrations, one with 25 and another one with 100 micromolar. So we use the 25 micromolar because it is the reported concentration found of these metabolites in circulation after the ingestion of fruits and vegetables. And we use the concentration of 100 micromolar because these are natural compounds. So probably they are associated with low toxicity. And at the same time, this ensures us that we are not missing effects derived of low compound concentration. Okay, so in this graph, we can see the neutrophilic inflammation index of the 29 compounds relative to the cholesterol inflammation control. Okay, so compounds with these values significantly above 1 are considered pro-inflammatory compounds, and the ones that have this value below 1 are considered anti-inflammatory compounds. And here we can see that 14 compounds have a strong anti-inflammatory properties at 100 micromolar, and 9 of them showed anti-inflammatory properties at the concentration of 25 micromolar. And so to proceed with our studies, we basically selected the compounds that were getting a stronger result at the lowest concentration of 25 micromolar. And as first, what we did was a dose response analysis of these topics so that we can find what are the strongest ones and also what is the lowest concentration at which they are active. And here we can see a good titration in some of the compounds, but also others that had, good titration in these two compounds, but also others that have an effect even at the lowest concentration of 5 micromolar, and thus they need further investigation to find the lowest concentration at which they are active. After these dose response analyses, we developed an assay that allows us to study intestinal function. Okay, and for this, we basically induced a chronic intestinal inflammation by feeding larvae with cholesterol for 8 days, and then we looked at intestinal peristalsis. And peristalsis can be easily analyzed with these chymographs, which basically represent the displacement of a defined area of the intestine in a certain period of time. And here we can observe the time between contractions in yellow, okay, and the contractions in blue of the intestine during 5 minutes. And as you can see, the normal fed larvae have several contractions, okay, that are basically delayed in the cholesterol fed larvae. And by quantifying this contraction frequency and the time between contractions, we can observe a nice phenotype where cholesterol fed larvae have reduced contraction frequency and also increased time between each contraction. And the idea now is to use this intestinal function, this intestinal assay, to test the compounds and to understand if these compounds can in a way improve the intestinal function of larvae with the inflamed intestine. And it will be also interesting to perform this in larvae with the neutrophils highlighted, so that basically we can correlate intestinal function and with the inflammation and the number of neutrophils inside the intestine. Okay, so as a conclusion, we have developed an in vivo discovery platform with Zebrafish larvae that allows us to screen the anti-inflammatory potential of several compounds by quantifying intestinal inflammation. Okay, then by testing common IBD therapeutics, we have proven that it is a platform that is suitable for inflammatory bowel disease related research. So then basically we used it to test a library of metabolites from which we have identified 9 with strong anti-inflammatory properties, which are very interesting for a more profound analysis of compounds' mechanism of action, for example. As for future research and for the most immediate future, we want to screen the entire portfolio of the 72 available low molecular weight polyphenol metabolites. We are at the moment trying to investigate how these topics can impact intestinal function and integrity. And for a more far future, we would like to understand the underlying mechanism of action of these compounds, of the ones that have given us a more robust result. And even for a future even further, we would like to validate the topics in higher vertebrate models, or even better in humanized 3D models of the intestine. Finally, I would like to thank all the tissue repair and inflammation lab, because it is an awesome laboratory, always with a good mood. It is very nice to work there, and everyone is very helpful. Then I will also like to thank Claudia Sanz and Diogo Carcosa for all the scientific discussion about low molecular weight polyphenol metabolites and for all of their availability, and also Telmo Pereira and Petra Pintado e Fábio Valério for all the help with the microscopes and with the fish. Yeah, thank you everyone for listening.
Thank you, Nuno.